Okay, good afternoon, everyone. So today we'll be talking about the second part of neuronal differentiation. So today's lecture is all about after they differentiated into neuron, what kind of structure they develop, what kind of protein they express. Because we know that you have certain progenitor for certain type of neuron. So you have expression of particular gene in the progenitor cells. Then they become mitotic. They become post differentiated and become post mitotic. And you have a different set of gene in the post mitotic neuron or the differentiated cells. Then after they become differentiated, they express certain. They have certain structure in the neuron that we are going to talk about today. It is really important to shape the neuron in a proper morphology. Also, it helps in the growth of the neuron, further growth of the neurons. So we'll talk about some of these important protein that is required for the neurodevelopment. So first, this is the initial stage of neurite outgrowth. So after the cell become differentiated or attain the certain fate to become different neuronal cell type, then this started as a rounded cell. So this is a fully this is a differentiated cells, and it has certain fate to generate particular type of neuron. So you can see that it's a differentiated cells become like kind of rounded. So initially it looks like a rounded cells. Then it shows some outgrowth projection from these spherical or rounded cells. So these are called as filopodia-like projection or lamellopodia-like projection that arises from these cells, and this is called as a neurite. The neurites. So you have generation of one neurite, then you have generation of second neurite in the opposite side. So you have generation of second neurite. Then after some some stage of the development, you have neurites coming out everywhere from this rounded cells. So they become become multipolar. So here they attain multipolar stage. So from this multipolar stage, one of the neurite, it develops into a long process called axon, and other neurite remain as dendrites. So we'll come to know what are the factors that regulate axon growth, or how the one neurite become axon, other neurite remain as dendrites. So we'll come to know that. So here this is a cell showing multipolar morphology, having many neurites, and you can see that these cells when they become a fully neuron, one of this neurite they elongated and become axon, and other neurite remain as a dendrite. And this is the cell body. So this image is taken in the bright field microscope, and here this is a immunostaining showing separation of dendrite and axon. So here the axon is marked with GFP, and the cell body, you have the nucleus here, and you can see that you have different marker, different protein that mark the dendrites. So we'll see those things in the coming slide, more detail. So if you look the structure of a neuron, so you have a cell body, then you have a process that arises from the cell body, that is called as axon, and you have also many processes arises from the cell body, that is called as dendrites. You have a long process called as axon, many small processes called as dendrite, and you have a cell body here. So we'll go over the anatomy of this neuron, how it looks like, what are the different structure that is present in the neuron. So here you can see that this is the axon. So here these are the important feature of the axon. So you have one axon per cell, and the important characteristics of the axon is you have mostly the microtubule which are oriented. You have the plus end oriented outward from the cell body. So you have plus end which is we can see that the plus. So this is the cell body, this is the axon, and you have the axon terminal somewhere here, which is not shown here. So the plus end is directed to the axon terminal. That is the key feature in the axon. The microtubule of the, you can see that the plus end is directed toward the axon terminal, and the major protein, microtubule associated protein in the axon is tau, which actually stabilize the microtubule. We are going to see that in the further discussion. So the major protein is tau in the axon, and the axon generates axon potential at the axon hillock. So you have somewhere here you have the generation of the axon potential that in the axon helps in the generation of axon potential, and the axon may be myelinated or it may be myelinated. You can see that you have a special type of cell which is encysting the axon for the efficient neurotransmission. So these are called as the myelinated cells. So you have the encysting of this the Schwann cells. So in between the two you have the roots run there. Also in the axon you have the microfilament. You can see that you have this microfilament which is also microfilament is also present in the axon. So this is these are the important molecules or important structure associated with the axon. So in the cell body, you have the nucleus, and these microtubules, they are randomly oriented. You can see that here the polarity is, the plus end is towards the axon terminal, but here they are randomly arranged. So you have plus end towards the cell body and the minus end towards the dendrite. It's not regularly arranged like axon you are seeing here. So you have the ribosome present in this cell body. You have a microtubule organizing center which organizes the microtubules. So you have many per cell dendrites. You have many per cells, and major protein. Major protein is microtubule associated protein 2, MAP2 in the cell body. The cell body doesn't generate axon potential, and they are non-myelinated. So these are the key feature of axon and cell body, because we are going to study the anatomy of neuron. Because after differentiation, what kind of structure you have, so that you have proper development or proper growth of the neuron. So we'll go some of these important key molecule like cytoskeleton or various cytoskeletal protein, how it is supporting the growth of the neuron. So different neurons exhibit diverse, different polarity. So here you can see that this is, you have cell body here, you have an axon here, and many processes from the cell body. These are called dendrites. So, and here this in the Purkinje cell, you can see that how complex it is. Even the cell body is small, and you have the axon here, and Purkinje cells, you have many dendrites. So, you have different, different cell have a different polarity, and there's so different morphology. There is a complex morphology among the neurons because that and different polarity. So here this is just showing that two different neurons derived from different region of the brain, mostly in the nervous system. So this is the spinal cord motor neuron. This is the dentate gyrus of the brain. So you can see that these are the different parameters, the measurement, how they varies from one cell neuronal cell type to other neuronal cell type. Here the cell body is around 8.4, and here is 0.4. So these are the different parameter. Kind of thought that it's not all the neurons, they do not show the same morphology. There is a variability among the different neurons, and the size of the axon, the size of the dendrite, the size of they vary among the neurons. That is what I want to say. They exhibit different pattern of polarization. So here this is the diagram. This experiment has been done to define the neuronal polarity in vitro. So we know that to study the neuronal polarity, it is very challenging to study in the in vivo model. So you can study the nature of neuronal polarity by culturing them outside in the lab. So this is a slide. This is the slide showing that how the growth of a neuron takes place and how they exhibit polarity. So first at 0.25 days, I think this is 0.25 days. So here you can see that, as I mentioned earlier, they first become rounded shape. They generate projection. Some of the projection are generated from this rounded cell called as lamellopodia or filopodia. You have generation of lamellopodia at 0.25 days. Then this lamellopodia develops into minor processes. You can see that these are the processes that arises from this cell body. And among all the processes, at 1.5 days, they become one process is elongated and become axon, and other process that develop into neurite, develop into dendrites. So these are dendritic outgrowth that develops from the cell body. And in the maturation, you have the cell develops into in 7 days those cells develop into mature stage. You can see that you have lung processes also arises from the cell body, and you have an axon. So this is how it starts with a small outgrowth kind of philopodia or lamellopodia-like projection from this spherical cell. Then they develops into many. They become multipolar. They attend multipolar morphology. Then among these, one of the neurites develops into axon, and other remain as dendrites. Okay, so not moving. Yes, so the axonal outgrowth takes place at 1.5 days, which is I have shown that here. So it takes around 1.5 days to one neurite become axon. Okay. Now, so there are some experiment has been done by other people to show that if you ablate or you do exotomy, you remove one of the axon, what happens to the polarity? So here there are a set of experiment has been done. So here the arrow that you are seeing here, this is one neuron, and this is the cell body, and this is the process of the axon. This is the axon, and these are the dendrites. So when they when they kill this one, they ablate this one, they look for certain times whether there is a development of another axon from the same area. So they have seen variability. There is a variability among the generation of axon. So here in this condition, the axon generated in the same area, so they attain the same polarity. But when they ablate this one, and they saw that this one grows into axon, so attain different polarity. In this condition, they again ablate this one, and they saw that this become an axon, like that. So you can see that they can attain the same polarity. They can attend they can regenerate. Actually, they can attain different polarity. Other dendritic process can become axon, depending upon you can do that in a live cell in a live cell imaging setup. So you can mark one axon, you can visualize you can mark one dendrite with a particular marker, and you can see that if you chop or if you remove one axon or the one axon, because you have one axon in one neuron, so if you remove that, the other dendrites, they try to become an axon. So that is what that is shown here. So they can attain the same polarity where you remove here, and this again regenerate to form an axon. They attain the same polarity. Here they are attaining different polarity. The other dendrites, they are regenerating as an axon. So that is what is showing here. So the different stage of axon development. So how this thing happens? This thing happens because of use of extracellular cues. That helps the cell body to polarize, that helps the dendrite to polarize to become an axon. So here this is experiment has been shown in a different time point with a different extracellular matrix. So they have taken two different extracellular matrix. This is the laminin, because the neuron for the proper development of the neuron, you need the matrix. So here the neuron can bind, and it can help in the it can extend its processes. So one of the substrate is laminin. Another substrate they have taken neuron glia cell adhesion molecule. So NG-CAM, the neuron glia cell adhesion molecules. So here what they have seen, when they cultured, they have coated the cover slip with two different substrate. One is the laminin, another is the NG-CAM. So you can actually take a cover slip or slide where you can coat the slide half with laminin, half with the NG-CAM, and you put these differentiated cells, which is fed to become neuron. So you can see that when they put this one, they generate the neurites. You can see that there is generation of neurites after 13 hours of plating the cells. Then when they cross this border, so this is the laminin and this is the NG-CAM in the same in the cover slip or cover glass. So when they cross the border, so you can see that these processes, that dendrites, they become as axon. So so the thing is that the neurites are responding to a change in substrate. So here there is a change in substrate rather than a specific substrate composition. So the same thing you can do also in another way. So you take you plate the neuron in the NG-CAM cultured cover slip or the slide, and you have a different substrate called as laminin, also coated in the same cover slip. So when you plate in the NG-CAM surfaces, when it touch the surface of laminin, this neurite become again the axon. So that means the neurite use the extracellular cues. So the extracellular cues here is, in this condition, the cues is NG-CAM, and in this condition, the cues is laminin. So they use the extracellular cues, to polarize themselves to become an axon. Okay. So this is called as a strip assay. So this is the strip assay that I have shown here. Then I am going to show another assay where the neuron polarize to become axon. So I am going to say the overlay assay where the neuron depolarize to become, to send its extent to polarize to become axon. So here, this is experiment has been done both taking in the lab. So they have taken this the cortical slice, which has a ventricular surface, cortical slice of the developing mouse embryo, embryonic brain. So here you can see that you have the ventricular zone, then you have a subventricular zone, then you have an intermediate zone, then you have a cortical plate, then the upper is pial matter. So you have this, the developing embryonic brain. So, and here this is the ventricular surface, this is the pial surface in the top. So what they have done is they have isolated, dissociated the cortical neuron, and they have labeled with a dye called as Deal dye, which can label the neurons. They want to see how the neuron behave when they cultured or plated those dissociated cells in this slice culture. So what they have done is they have labeled the cells with this dye, and they have plated here. So you can have two scenario. Scenario one, you can have a random growth of this the plated cells in the in this cortical slice. So you can have extension of you have the cell body. You can the axon can extend in the upper side. It can go lower side, anywhere in the cortical slice. The other scenario could be these cells, they're extending their axon down to the ventricular zone. That could be one possibility. And here they have seen that in actually the axon is the neuron is extending their axon towards the ventricular side. That is because of the semaphorin gradient. So in the cortical slice, you have this morphogen called as semaphorin 3A. So which is a repulsive molecule that repels axon growth. So what happens is, so you have more gradient of semaphorin in this region, like in the pial side, the upper side. That's why the cell body, that's why the axon is not going in the upper side. That's why it's going in the lower side, because you have lower gradient of because we are culturing that plated cells in the cortical slice, and in the cortical slice you have higher concentration of semaphorin A in the upper side. You have a lower concentration in the ventricular side. That's why the axon is responding to these signals. So axon is responding to the semaphorin signal. That's why you have, you can see that in this real image, you can see that all these the plated cells, their axon is going downward, along the semaphorin gradient. So you have less semaphorin gradient in the ventricular side. That's why the axon is extending their projection towards the ventricular sides. So that is how you can see that the semaphorin A, how it is polarizing the neuron, to a different region, the ventricular region. Okay. Any question so far we have discussed? Okay. So yeah, today class is pretty simple because we are going to see, we are not going to discuss any genes and all these things. We are going to see after they become differentiated, how they behave and how they grow, and what are the structure that is actually regulating the growth of the neurons. So among all these cues, extracellular cues, or to attain the neuron in a different polarity, you need different proteins to for the growth of the neuron. So here I am highlighting 3 different protein that is present in the neuron, which is actually giving a framework, which is actually shaping the neuron. So one of the most abundant protein is the microtubules. So here the microtubule is around 20 nanometer diameter. So you have the neurofilament, you have the microfilament. So these are the cytoskeleton which provides support to the neuron, the growing neuron. Okay. So we'll talk about first with the microtubule, how they are formed, what are they, and the neurofilament and microfilament. So the microtubule consists of two, so they grow like, so you have two different ends of the microtubule. One is the growing end, this is the polymerizing end, and this is the depolymerizing end. So it grows in the plus end. Plus end is the polymerization end. So your alpha, so the microtubule consists of two distinct kind of subunits or tubulin subunits. One is alpha tubulin and then the beta tubulin. So alpha beta tubulin, they make heterodimer and they polymerize at the plus end. This is the growing end of the microtubule, and this is the depolymerizing end. So it looks like this. The structure of microtubules, and they provides this support during the growth of the neuron. Okay, so the polymerization depolymerization are inherently asymmetric. So, and on the polymerization mostly happens in the plus end, and depolymerization happens in the minus end. Okay, and the depending upon the condition, the cell can polymerize and depolymerize the microtubules. It depends on the cell where it wants to polymerize or depolymerize. And mostly these microtubules or that you are seeing here consists of beta and alpha tubulin. They are more stable when they are associated with other assisting protein called as microtubule associated proteins. So we are going to talk about some of the microtubule associated protein and how the microtubule associated protein is helping to stabilize the microtubule subunits. Okay, so mostly the microtubules are found in the axons. They are very stable. They are also found in the cell body. So I'll be showing in the coming slide. So this is how the microtubule looks like. So you have a kind of a cylindrical structure which is actually providing the framework in the developing neuron. So here these are the subunits of the microtubule, alpha tubulin and beta tubulin. And you have, this is the minus end, and this is the plus end, this is the growing end of the microtubules. And as I mentioned, that microtubule is always supported by the associated protein to provide a framework that is called as microtubule associated protein or MAPs. Okay, so the MAPs are two. They are the structure of MAPs is, so MAPs are high molecular weight MAPs. So you have so we'll come to these points, but I want to just highlight that this microtubule associated protein, you have two ends. One end is binding to the tubulin subunits, the alpha and beta tubulin, and the other end is the projecting end, which is binding to another microtubules. So that's why I'm saying that this microtubule associated protein, the MAP2 or MAPs, they helps in bridging this among the two microtubules. So this is one microtubule, you have another microtubule here, or another kind of other structure here. Actually, it is providing a, like in the building you have, if I show like this in the building, you have this pillar and it is cross-linking and it's providing the mechanical support. That's why you have stability in the building. The same thing in the cells, in the neuronal cells, the microtubule provides stability to the axon, provides stability to the cell body or dendrites. So one end is binding to this tubulin subunits. Other end is binding to either to the microtubule or any other structure. Okay, so it consists of the MAPs. It consists of the high molecular weight, mostly high molecular weight MAPs, mostly present in the dendrites. The cell body, you have the projection coming out from the cell body called as dendrites. Those are the high molecular weight MAPs. And another MAPs, and that is called tau, that is present in the axon. That is also presenting, helping the axon to support. So the high molecular weight MAPs present in the dendrites and the tau present in the axon. These are the microtubule associated protein. Either the MAPs, the high molecular weight MAPs, or the tau. Tau in the axon, high molecular weight in the cell, in the dendrites. Okay. So the tubulin binding end facilitates actually it stabilizes and facilitates. I mentioned that stabilizes the microtubule. Okay. So these are the important key feature of the microtubule associated proteins, and it provides kind of support cross-linking one microtubule to other microtubule, and also cross-linking one microtubule to other structure like vesicles, so that so that you have proper function of the neuron. So here, this is an electron micrograph showing cross-linking between the two microtubules. So these are the structure that you are seeing here. These are the vesicles, synaptic vesicles, and here the arrow that you are seeing here, that is it's not it is difficult to see in the electron microscopy, but that they have mentioned that the arrow that you are seeing here. So this one microtubule is cross-linking to other microtubules, and the star that you are seeing is the microtubule cross-linking with the vesicles. So you see such kind of arrangement in the cells, neuronal cells. That's why you have the proper development of the neurons. So so another protein that also plays an important role, providing the framework, the support in the axon, is the neurofilament. So we are going to talk about the neurofilament. So microtubule is around 20 nanometer diameter, consists of alpha tubulin and beta tubulin, along with microtubule associated protein. And the neurofilament is around 10 nanometer in diameter. So we're going to talk about some feature of the neurofilament. So the neurofilament is, you can see that the neurofilament also provides strength and stiffness, especially important in the long axon. So you have the axon. So axon have this neurofilament, which can actually the long axon are having this neurofilament, and that is actually supporting the axon to develop. So you can see that this is forming the mesh-like structure and is supporting the axons. So you have also some neurofilaments are also present in the dendrites. Okay. So they are the principal structure which support the axon. So axon mostly supported by neurofilaments. Okay. The same thing I mentioned, that it provides strength and stiffness, especially to the longer axons. So this is how the neurofilament assembled, and it forms a meshwork, like forms a filament like this. So it starts with a polypeptide chain. So here you have the N terminal, you have the C terminal tail. So these two polypeptide chain, they from the dimer, they join N to N, C to C, and they, again this dimer, they form tetramer. Okay, they form like this. You have N to C, C to N arrangement. And the tetramer, they again arrange to form the protofilament. So they form one protofilament, around 6 tetramer, 6. So you have one tetramer here. So 3 tetramer, they form one protofilament. Then the protofilament, they arrange themselves to form the filament. So this is how the microfilament is formed. Just want to show that how they looks like, and what is how they are assembled together in the cell to become the filament. So this is how the filament looks like, neurofilament. Then this neurofilament actually provides the 3-dimensional structural stability to the axon. So here you can see that the axon is supported primarily by neurofilament, and they are cross-linked to each other, and there are high molecular weight neurofilament also present in the axonal compartment. So the dendrite primarily consists of the microtubule that I mentioned before, like the microtubules, and also dendrite also consists of some neurofilament, very not that enriched, but yeah, there is also presence of neurofilament in the dendrite, but mostly they are enriched in the axon to provide the support to the long growing axon. Okay, so just remember that axon is highly enriched with neurofilament, and the cell body, the dendrite, is mostly associated with microtubule and microtubule associated protein. Okay, so now this is a cartoon showing how they are organized. So you have here the organelle. So these are the microtubules present in the cell. So these are the structure that you are seeing. These are actin-rich structure. These are the microtubules. So that is how they looks like in the cross section of the axonal, in the axons. So this is the same thing. I'm showing that how the microtubule, they cross-link among themselves to support the axon, or to support the dendrites. The microtubule associated proteins, same thing. I'm showing that the cross-linking, they provide support to the to the growing axon. Okay, so the microtubules So let us talk about some of the microtubule associated protein and how the microtubule associated protein helping in the axonal transport. So this is a cartoon showing a structure of the microtubule consists of alpha and beta tubulin. So you have the plus end, the growing end, the minus end, the depolymerization end. So the depolymerization happens here. The loss of my alpha and beta tubulin happens here. And this microtubule associated with two sets of protein, motor protein, which is regulated by the ATP. So this is kinesin protein and dynein protein. Kinesin protein actually takes cargo from cell body to the axon terminal, cell body to axon terminal, kinesin, the anterograde transport. And dynein protein takes cargo from axon terminal to the cell body, retrograde transport. Okay, so kinesin towards the plus end means towards this axon terminal, and dynein towards the minus end, that means towards the cell body. Okay, so they help in the transport of different organelle, different protein, different signaling molecule. So with this microtubule associated protein, so the microtubule gives support to the neuron, and microtubule associated protein also gives support to the neuron. Along with, they helps in transport of certain cargos, like the kinesin helps in transport of cargo from axon terminal, from cell body to axon terminal. That is called as anterograde transport. It helps in the anterograde transport. Okay, I might ask question: what is the role of kinesin? The anterograde transport, retrograde transport. You have to remember this. It takes cargo, from cell body to axon terminal. That is called anterograde transport. And dynein takes cargos from axon terminal to cell body. That is called retrograde transport. Okay, so kinesin and dynein. So most of the axonal transport, what happens is, why axonal transport is required? Because most of the protein that is synthesized in the cell body, you have the nucleus in the cell body, and you have long processes, the axon terminal far away from the cell body. So you need transport of material from cell body to the axon terminal. That's why you need this motor protein, either dynein or kinesin. Mostly the kinesin helps in transport of material from cell body to the axon terminal through the microtubules, because the kinesin can walk along the microtubule, like it can walk along the microtubules. So here, this is the cell body here, and here the axon terminal. So this is the kinesin. It goes like this. So it can take cargoes from cell body to the terminal, and the dynein also can take cargo from terminal to the cell body, because you need certain signaling molecule that should be transported from the terminal to the cell body. It should give information to the nucleus, so that nucleus will transcribe the gene. Okay, so the dynein does this. So here I'm just showing that this axonal transport is important for transport of a cargo, for transport of cell organelle, like to the different part of the neuron. And also this suppose one neuron gives signal to other neuron. How this happens? This happens because of this. One neuron is giving signal, so you have the transport of these things through the microtubules, or just showing this example here, the reflex axon. This is again the transport of signal from one neuron to other neuron. So here, suppose you are walking along the street, you just encountered with a very sharp things, right? You put your foot in your sharp thing, then you take out your foot. This is called a reflex axon. This is happens. This happens because of the involvement of spinal cord. So here, what happens? Is, so you have the leg is here. So it gives. So you have this muscle spindle here. So there is special type of neuron which is connected to this region, and it sends this your stimulus, and it gives signal to the spinal cord. So here the brain is not involved, because it takes a lot of time to process the signal to the brain. The brain will communicate. So the reflex action does by the spinal cord. So here, this neuron is communicating. This neuron is you have this sensation that is sensed by this neuron, which is which is here. This is called as afferent neuron, and it gives signal to this to the spinal cord. And you have another neuron in the spinal cord as alpha motor neuron. So here, the signal is transported from here to this, and in the axon terminal, and from the axon terminal, this is called as anterograde transport. Okay, cell body to axon terminal. Then So here it is giving signal to here. This is axon terminal. Here the you have cell body somewhere here. So it is axon terminal. So then this is again communicating with another neuron. So this is called as alpha motor neuron, and this is giving signal to the muscle. Then the muscle contract and you take out your leg. So that is happening because of the processing of signal from sending like sensing of signal from one neuron to other neuron. That is because of this involvement of this axonal transport, the fast processing of this signal. So that happens because of the retrograde transport from one neuron to other neuron by dynein protein. Okay, so that is one to show that this thing is really important. The communication between one neuron to other neurons is really important, and that is happen because of the involvement of the motor protein, the different types of motor protein like kinesin and dynein. So here this is showing how transport happens along the microtubules. So here this is the axon and this is the cell body. These are the dendrites. So you can see that in the axon, those these motor protein, the kinesin, they are oriented towards the plus end. So you can see that they are going towards the plus end. So they take cargoes from the cell body to the axon terminal, and the dynein protein that you are seeing in the blue, they takes cargoes from the synapse or the terminal to the cell body. So this kind of arrangement you have in the axon, but you don't have this kind of arrangement in the dendrites. So they are mostly very specific. The transport thing happens very specifically in the axon terminal or in the axons. That is one to show. So the same thing I have just represented here. So you have a cell body here, you have a terminal here. So this is the axon. The kinesin actually taking signal from cell body to the terminals, and the dynein from the terminal to the cell body. Okay. So in axon, kinesin moves vesicles and other organelles. So kinesin can move the vesicles, synaptic vesicle or other vesicles, from cell body to the terminals toward the axon terminal. But dynein takes cargoes from terminal to the cell body. That is what I want to highlight. So here are some of the important features in the axonal transport. The fast axonal, the axonal transport can happen very fast, very slow, depending upon the requirement. So the fast axonal anterograde axonal transport, that is done by the kinesin from the cell body to the axon terminal, and that happens around 400 millimeter. It can move the vesicle 400 millimeter per day or 300 microsecond per minute. So mainly the organelles or the vesicles are transported by fast anterograde axonal transport, and the fast retrograde axonal transport can also happens. So, and this retrograde transport happens when the neuron tries to send signal from one neuron to other neuron. Like you have the axon terminal, you have the cell body. Suppose you have another neuron which is also communicating to the axon terminal. So it gives signal to the axon terminal. Now the axon terminal will send signal to the cell body. So that is called as retrograde transport. So mostly the fast retrograde axonal transport mostly carries the signaling molecule from axon terminal to the cell body. Okay, and also you have slow axonal transport mostly by the actin molecules are transported by slow axonal transport, other metabolic enzyme like glycolytic enzyme or enzyme required for the neurotransmitter synthesis, that is happens by slow axonal transport, or the slowest axonal transport mediated by the microtubules or the neurofilaments. Okay, so you have the axonal transport because to proper function of the neuron, you need this kind of feature, you need this kind of activities in the neurons. So here again showing that the different thing here, like how this you can stabilize the microtubules by using other pharmacological agent. So here this is hippocampal neurons in the cultured conditions. So here you can see that this is the cell body, and you have various processes arises from the cell body. These are called as neurites. Okay, so you have 10 neurites, like neurite 1, 2, 3, like is labeled as 10 neurites, and this neuron, hippocampal culture neuron, is cultured with a pharmacological agent called as taxol. Okay, that is called as caged taxol. So these are cultured with the taxol, and taxol actually stabilize the microtubules. Okay, the caged the caged taxol, it doesn't stabilize the microtubule. On-caged taxol stabilize the microtubule. Okay, so on-caged taxol actually stabilize the microtubules. So here the neuron is cultured with a taxol, caged taxol. Okay, so you can actually, we want to know what happens if you uncage this taxol in particular neurites. So here this neurite is just labeled. So we are marked this neurite with this rounded kind of. They have marked these neurites, and they uncage this taxol by putting the UV light. So they grow these cells with caged taxol. Now they want to study the development of one neurites. Okay, so they photoactivated that neurite by putting the UV light, so that the taxol here, which was caged, become uncaged. So here they become uncaged. So you can see that after 11 minutes of uncaging the taxol, you do not see the growth of the, you do not see any problem in the growth of the microtubule. You can see that this 0 minutes you have the growth of the microtubule. After 11 minutes, you still see the growth of the microtubule. That means uncaging the taxol doesn't affect the microtubule growth. Okay, so what they have done when they want to stabilize the microtubules and want to see whether the stability of the microtubule helping the neuron to grow much better. Okay, so the caged taxol, just remember the caged taxol, it is not a good condition to stabilize the microtubule. You have to uncage the taxol so that it can stabilize the microtubule. So they have specifically uncaged the taxol in these neurites. And after uncaging, they see that there is no change in the microtubule, microtubule structure. So they found that when they uncage the taxol in this specific neurite develops into axon. That means there is more growth of the microtubules. That's why you have development of among all these neurites, one develops into the axon. So you can see that again they have marked with the two different protein. So the tau mostly labels the axon. So you can see that this become axon, and other processes that you are seeing here, which is already in a caged condition, they become the neurites, the dendrites. So you can see that when they uncage that, so you have more development of the neurites. You have the neurites developed into the axon. That is what is shown here in this slide. Okay, so here this is a movie that is showing. Just play the movie, and how the kinesin, how this is a kinesin motor protein, that is present in this dendrite that is transported to the axon. So you can see that. You can see that this is with time this come to the cell body from the axon. So this is the anterogate, just to showing that anterogate transport, from the cell body to the axon terminal. So it was here initially, then it come to the cell body, localized to the cell body, then here. So it is anterogate transport. Okay, so kinesin motor protein initially localizes to all the neurites, then they travel to the cell body, and from cell body they again travel to the axon terminal. So the kinesin mode, this experiment suggests that in the kinesin is a anterogate transporting molecules transport the cargo from cell body to the axon terminal. This is the axon terminal. Okay, so the same thing that is just showing with time. So then the neurites that we are talking about, you have formation of a neurite, many kind of filopodia-like or lamellopodia-like projection. Then one neurite is becoming the axon, depending upon the external cues that I have shown before. They require this semaphorin signal or the external cues, the different substrate. They cross this, when there is change in the substrate like laminin or NG-CAM, in the NG-CAM, that neurite become the axons. So how the neurite elongation happens? So the neurite, so here this is the cell body, then this is the neurite, one neurite, and this is the growth cone of the neurite. So the elongation of the neurite happens at the growth cone. And this growth cone is actually enriched with actin-rich structure. These are actin-supported extension of a developing or regenerating neurite that is seeking for synaptic target. So this structure, they want to, they want to communicate with the other neuron. So this structure that you are seeing here is one neurite, and this, this tip of the neurite become growth cone enriched with actin and other cytoskeletal proteins so that they can grow and contact to another neuron. So this work is done by initially work was done by the Kahal, and he found that the tip of the neurite is called as he found that the tip of the neurite is enriched with actin-rich structure, helping in the growth of the neuron. Okay, so here is showing that different developmental stage of the neuron, showing the how the neurite grows to form the axon. So here this is you can see that this is 18-hour, 19-hour, 20-hour. This you can see that the neurite elongate, and in the growth cone, this structure is the growth cone. They become long axon. So you can see that the growth cone, like the growth happens at the growth cone of the neurites. Okay, so neurite elongation occurs at the growth cone. So we'll talk about how the how the neuron grows at the growth cone. So this is the typical structure diagrammatically shown here. So this is the axon, and is the axon terminal, the growth cone. So in the axon you have these microtubules, you have the microfilaments. So and they grow in the microtubules. They grow towards the you have minus end here, you have plus end here. So they grow in the plus end. Along with in this terminal region, you have other cytoskeletal protein called as actin cytoskeletal protein, which is also helping in the growth. So what you can do, so the people have done certain experiment to ablate or to remove or to disorganize the microtubule in the axon, and want to see whether the growth cone development is affected. So here what they have done is they have bleached this the microtubule that you are seeing here. With time, the bleach area is again regenerated by the turnover by other tubulin molecule, because you have addition of more tubulin molecule towards the plus end. But even after removing this tubulin or the in the middle of the axon, you do not have any problem in the growth cone, because you have other structure at the tip of this growth cone that is called as actin, which is helping growth of the axon. So the tip of this growth cone is the tip of this neurite is enriched with the actin, and that is helping in the growth of the axon. So here and these are mainly consists of the microfilament. These are the actin-rich structure in the growth cone, the microfilaments. The third thing that I want to highlight is the microfilament and the growth cone. So here you have two different pools of actin called as G actin, the granular actin, and the F actin. So the G actin, the granular actin, they polymerize to form F actin, the filamentous actin, at the tip of the growth cone. Now the growth cone grows because of this polymerization of the G actin to F actin. Okay, so the polymerization happens, the G actin from the F actin. So here this is the typical structure showing the growth cone structure. So the growth cone of the axon terminal enriched with mitochondria, because it requires ATP. So that's why you have the local availability of the mitochondria, because the polymerization of G actin to F actin requires ATP. So that's why the mitochondria here. Now you have actin filaments also. These are the actin filaments. You have the microtubules, mostly in the axons, because it transport the cargoes and the other cell organelles. And the tip of this neurites consists of the actin-rich structure. Okay, and you have generation of this growth cone and growing of the neuron. So this is just showing that this is just another model showing how the growth happens at the growth cone. So you have these are the microtubules, because you need two distinct things for the growth of the growth cone. You need the membrane, because you have addition of membrane. Also you need the actin. That's why the neuron can the membrane can grow, or the growth cone can grow. So the microtubule, what it does here, the present in the axon, they transport the vesicles, because you have the vesicles, the new membrane, okay, the vesicles, and in the tip of this growth cone. And also you have polymerization of G actin, the granular actin, to the F actin, the filamentous actin. So you now you have the transport of the membrane, you have the F actin. Now this grow added in the tip of this growth cone, and the neuron grows like that. So that is how you have that is how the neuron grows, because of the development of the growth cone. So this is the staining showing that how the growth cone looks like in the real scenario. So here the growth cone is labeled with the F actin after the formation of this filamentous like structure at the growth cone. So and this microtubule is in the green. So you can see that the microtubule mostly present in the axon, and some also there in the very less there in the terminal, but mostly in the axon. And the axon terminal, the growth cone is enriched with actin-like structure, the filamentous actin-like structure, which is basically providing support and also helping in the growth of the axon terminal, Okay, so this is all about differentiation. So you have generation of progenitor cell. Progenitor cell express different sets of gene. Then it become mitotic. Then it differentiated to become after post mitotic. You have expression of different sets of gene. Now you have development of other structure that helps in the growth of the neuron. So this is how neuron originated and grows to become a mature neuron, functional neuron. Okay, so this is all about the differentiation. Any questions? Okay, so I'll talk to Ryan regarding the grading thing, how this can be done. You can see. So I'll talk to him. Thank you for today's class. So we'll be talking next class will be about synaptogenesis.